is Stephanie Green. Of course, that's the future. Oh, and I am also supposed to announce that following Stephanie's talk on April 11th, if you can just all dutifully walk together as a pack over to the Lougheed Center, we'll be having the department's Celebration of Excellence right after the Chair's Lecture Series at 4:30 over at the Lougheed Center. But today, it is my pleasure to be able to introduce Dr. Kirst King-Jones and to present him with his department's swag for the effort that he's taken in putting together a presentation this afternoon about his research. Kirst, the floor is yours. Yes, and it does come empty.
Thank you.
Is it made of iron?
It is some kind of metal. Yeah, so I just talked to Mike. It would be better if we fill this up next time. Double-blind test or something. Anyways, thanks, Mike, for the intro. And thanks for coming. I didn't know how many would show up. And I realized when I prepared this talk that this is the first time I actually have a drink during my talk and where the audience has access to alcohol. And so I thought, probably the main reason you're here is that 99% that Mike mentioned, and not so much my talk. And so then I realized, well, perhaps it would be a great idea to make this more widespread. When you go to a scientific meeting and there would be like a drink advisor, and you would go to them and say, hey, so what's the next talk? And they would say, it's this German guy, he has a heavy accent, talks about iron and genetics, so I would recommend this drink here. And so I thought, because Mike, is a wine enthusiast and knows a lot, probably the cocktail. What do you think that appropriate drink would be for today's presentation?
I would say a fine Burgundy, probably from around.
A Burgundy. That's not bad. Not bad choice.
A good Pinot Noir.
How do you mean that's not bad?
It's not bad.
It goes perfectly with
Burgundy Noir.
Yeah. Well, thanks for your input, but those are all incorrect answers. So I'm going to show you later in a slide what the correct answer is. All right. So perhaps I should get started about what I'm going to talk about today. So basically, historically, my lab has been interested in the study of steroid hormones and how they are synthesized and how this process is regulated. And that led us to become interested in iron because there's a connection between steroid hormones and iron. And I hope this will become clear in the next few slides. So this basically represents the work from the last 3 years or so. And when you think about iron, it kind of makes sense that this is incorporated into living organisms and ubiquitous because it is one of the most abundant elements on Earth. And so it's no surprise that humans have 3 to 5 grams in our bodies. And so, of course, this means a little part of us is an iron man or an iron woman. So, and this, of course, begs the question, what do we need iron for, right? And the answer is that we require iron for 2 types of protein cofactors. And one of them is the famous heme molecule, and you can see here iron at its center. And it's most commonly bound to hemoglobin to transport oxygen. And one of the lesser known protein cofactors that contains iron are iron sulfur clusters. And they look a little bit like this. They come in various formats, and their production is very complicated. So in part, they're made in the mitochondria, and then they're polished off in the cytoplasm. And while we're at this iron slide, lots of the questions that are still open revolve around these 2 cofactors. So to this day, we don't know how iron and how a heme is transported in the cell, for instance, or how iron is transported into mitochondria, and how this compound here, this cluster, is properly assembled and inserted into so-called client proteins. So there are many questions we don't know about the cell biology of iron. So the examples I want to mention here, and they come later in my talk, are the aconitase and cysteine. So I will come pretty quickly to the aconitase and then talk about this other protein. So these are examples. But there are literally hundreds of proteins that require either heme or this cofactor, and they need this to function and exert their biochemical reaction. So why is iron so ubiquitous? And it all boils down to its amazing property to donate and accept electrons. And that makes iron so versatile in a wide range of biochemical reactions. But this same property that makes it so useful also makes it very dangerous. So if you have too much iron in your cells, you will actually likely produce so-called radicals, for instance, reactive oxygen species, and those are cell damaging, and they can damage cells and DNA and wreak havoc. So the take-home message from this is that iron levels have to be strictly regulated both on a cellular level and on an organismal or systemic level. Now today's talk focuses on how cells regulate their iron and not on an organismal level. And vertebrate cells, actually plant and metazoan cells, do this by using a protein called iron regulatory protein 1. And so this is one of the main topics of my talk today. But we won't start right away with that. I will first tell you the story how we got there. I should mention that there is a second protein in humans, which I'm going to ignore for the most part, called iron regulatory protein 2. So let's dive right into this central dogma of iron biology, and which I call the central paradigm in my title. And this states that cellular iron regulation works as follows. So you have this IRP protein, and when iron levels are normal, it actually harbors this iron-sulfur cluster, and this protein therefore acts as an aconitase, and that's an enzyme that interconverts citrate to isocitrate. So it's a reaction and can go in both directions. But when iron levels drop, this protein changes its conformation and now becomes RNA binding. So it can bind to very selected messenger RNAs, shown here, that contain these so-called iron response elements. So these are little stem loops, and they are recognized by this protein. And the net outcome of this is that more iron is imported and less iron is stored. And overall, iron availability increases in these cells until levels go back up. So that's sort of the central paradigm. And when you ask people at iron meetings, so what is the aconitase for? What's the importance of this? You get a shrug and people say it's not important. And the reason for this is that there is a mitochondrial aconitase that works in the Krebs cycle, or a citric acid cycle, as it's also known, to basically produce energy for the cell. And so people think that the citrate will enter mitochondria, and that's pretty much it. It's like a little bit of an extra level of citrate. And so today I want to convince you that that is completely wrong, and that not only that this aconitase is essential for viability, at least in Drosophila, but also has a role in iron homeostasis, which was very surprising. And so this paradigm has featured the textbooks for more than 30 years, and both Mike Harrington and I have taught this in Gen X 3 or 4. So how do we get to this point? And I sort of want to backtrack a little and introduce our beloved fruit fly to you, our system we're working on. And so here it is. So you have seen this probably many times. And I want to point out that we're actually working on larvae, so I want to show you briefly the life cycle of the fruit fly. And I want to also connect this right away to these steroid hormones I talked about in the initial slide. So here's your life cycle of the fly. It takes roughly 10 days from the embryo to the adult form. There are 3 larval stages. And Drosophila is unique in the sense that it's the only model organism where we have mapped the corresponding steroid hormone titer changes throughout development. So it's quite a useful tool to understand what's going on. And you can see that the molts from a larva to the next stage are always preceded by a major pulsific diazone. And for instance, pupariation, the beginning of metamorphosis, is also preceded by a huge pulse. So a diazone is the principal steroid hormone in insects, and it drives development. It acts sort of as a timer to coordinate multiple events to drive development. And so we are working mostly on the third instar. And so in the next slide, that shows you a bit more detail on the system we're working on. So when we zoom into this anterior part of this larva, you can see the larval brain and the tissue we're working on and are very interested in is this colorful ring structure here that I drew in here. And this is an organ referred to as the ring gland. And in diptera flies, it's a composite endocrine gland of 3 glands, namely the corpus allatum, the corpora caudiliaca, and the prothoracic gland. And that's what we need to focus on, the red part here. That's the prothoracic gland, and that's where steroid hormones are produced. Now, when you stain this tissue, this gland, for iron, you'll notice that in this dark stain, that it's much darker stain than the 2 neighboring glands here. And further work basically showed that, from our lab, that the prothoracic gland is iron-rich, and that it probably contains more iron than most other tissues in the fly. And so why is that? And I promised you there's a connection between iron and steroid hormones, and so here it is. So whether it is in plants, in humans, or in insects, all steroid hormones are produced from suitable sterile precursors, and the most common one would be cholesterol. And so cholesterol is then metabolized in multiple steps to the mature hormone like testosterone or ecdysone in insects. And most commonly, or the most commonly used enzymes that do these reactions are the so-called cytochrome P450s. And they only work if they have a heme group bound to them. So, and heme, of course, contains iron. So if we take a closer look of what's happening in the fruit fly, we are dealing with a pathway that's pretty well understood. And these here are the enzymes with these strange names called Neverland and Shroud and Disembodied that essentially convert cholesterol to ecdysone. Now, shown in red are the enzymes that require iron in one form or another. And if we start with Neverland, you can see that this is an iron sulfur cluster protein, while the last 6 enzymes are all cytochrome P450 enzymes. And because these are mass products, you require massive amounts of heme and iron in these prothoracic glands. And not only that, it has to be also dynamic because ecdysone is released in pulses. So there's some interesting cell signaling or regulatory pathways going on that determines how much iron is released or used at a certain time. All right, by the way, you can disrupt me anytime and ask questions if you feel like, what was that? Or what was that German thing? All right. So to understand how we approached all this, we need to first take a look at how heme is produced or synthesized in animals. And this looks overly complicated, but I'll try to explain this briefly and point out the important things here. So the 2 precursors that are converted in the first step by an enzyme called ALAS to a compound called ALA, and that's shown here. And the trick really is that multiples of this ALA are then used to form probably the most extravagant molecule in biochemistry, and that's the tetrapyrrole. And they're called extravagant because they are purple and have the ability to fluoresce in a bright red. I'll get to this in a second. So this ring structure then is then further converted into the mature heme. In the final step, iron is incorporated and you get mature heme. So I mentioned this fluorescent ability. So when we take a look at these precursors, number 4, 5, 6, 7, I have drawn them here in red, indicating that they have the ability to fluoresce. And so these correspond to these precursors with a ring-like structure without iron. Okay, in a normal cell, we basically, well, I should say that this fluorescence only occurs under oxygen and in UV light. So you would, if you take a normal cell and basically expose it to UV light, you wouldn't see anything because these precursors are very short-lived and are rapidly converted to heme. But let me show you what happens if you disrupt this process at various stages, namely here at step 7, 5, and 4, I believe. Let's take a look. So here, we are using a technique called RNA interference. And basically, it's a way to impair the gene function by disrupting the messenger RNA that's produced from a gene. And we can do this in a tissue-specific way. So what you're looking at are the dissected brains with the attached ring gland under normal light and under UV light. And this is a control, and as I promised, there's no fluorescence in a normal control animal. But when we disrupt the process, we get this beautiful red fluorescence, and also an enlarged ring gland in many cases. So that is such a strong phenotype that you don't even have to dissect these tissues out. You can see this, in fact, in an intact larva. Okay? So here we have, again, a specific disruption of the gene only in the prothoracic gland. So what happens if you do this in the entire animal? This would be a classic mutant where every cell is affected. And this is how it looks like. Here we're disrupting PPOX. That's the second last step in the production of heme. And you can again see the ring gland and two other tissues called the enocytes in the gut, which we believe also produce high amounts of certain cytochrome P450s. So we believe that these three tissues here, gut, enocytes, and the ring gland, all are iron-rich tissues with obviously high iron demands. Now this sort of accumulation of heme precursors where heme production gets stuck is actually something we know from a certain group of human diseases known as the porphyrias. And the porphyrias is sometimes also commonly referred to as the vampire disease. And that's because in its most severe form, which is Gunther's disease, which I think affects step 4, the gums retreat and the teeth look longer. And because UV exposure makes everything much worse, you get these terrible skin lesions. These patients tend to stay inside and come out at night. And importantly, during an acute porphyria attack, you can actually cure this by an infusion of heme today, but in the old days, you could drink blood. So people believe that this could be the origin of the vampire folklore, but of course it's not really proven. So you can see here also this fluorescent accumulation. All right. So we thought we can use this fluorescence that I showed here and in the last slides here to conduct what we call in Drosophila genetics a genetic screen. So what we're going to do is we're going to disrupt thousands of genes, and we're going to look for autofluorescence under UV light and hope that we can find new genes important for the regulation of iron or heme metabolism. Okay? And so here's what we did. So again, we use the same RNA interference approach where we can disrupt gene function in the prothoracic gland. You don't need to really know more details. And we screened 12,500 genes. So this is quite a monumental effort. And so we split this work among 3 labs where we screened roughly 4,000 lines in the 4 months in summer. So there were roughly 20 people involved in doing that. But our initial interest wasn't this. At that time, we didn't know about this. But we wanted to find developmental defects where, for instance, metamorphosis wasn't occurring. So once we had identified roughly 1,200 genes, every lab then worked with their own interest and follow up on that. And what we did in our lab was taking roughly 800 lines where the animals would develop as far as to the 3rd instar, so that's the last larval stage. And we would look for these enlarged, red, beautiful autofluorescent ring glands and let the animal tell us which genes are important for heme and iron regulation. And to make a long story short, we found 34 genes which we dubbed the bloody genes. Okay. And these genes really ranged from transcription factors, so regulating of genes, cell signaling, lysosomal genes, transport genes, so a whole cool range of cell biology, and we still, of course, are working on so many of these genes. But today I will be talking about a gene called AGBE. Okay, and before I tell you what AGBE is, I want to give you the answer, what the appropriate drink is for this talk. All right, so we talked about Halloween genes and bloody genes and vampire folklore. So the red wine was not a bad suggestion, Mike. I could give you half a point for trying. But the correct answer was Vampire vodka. That is the correct answer. All right. Never heard of this stuff before. Had to Google it. But, okay. No, I should have brought a bottle. Like whoever gets it right gets this bottle.
Sangre de Toro.
All right. So I just mentioned this new gene that we got out of this screen. So it's called AGB. Let's take a look. First, what we found. And so here's what came originally out of the screen. And this is what happens when you disrupt heme biosynthesis directly. So you can appreciate that this looks very similar. And the controls, again, show no fluorescence. Now, the problem with this RNAi technology is that it often is unspecific. And you want to make sure that this is real. So what we did is get another line that works through a completely independent means, because it basically targets a different part of the gene, if you wish. And that showed you the same phenotype. So we felt pretty good about it. But I have a deep distrust against this technology because we had problems in the past. So we didn't want to stop there and used newer technologies, namely CRISPR-Cas9, to confirm these results. And so here's what we did. So I think we use probably 4 different flavors or variants of CRISPR-Cas9 in the lab. And this is the most complicated one you can actually do. And it's, in a nutshell, what it's doing is it's replacing the original gene with something we modified. Okay, it's called a knock-in. And we modified it to the following specifications. So first of all, we introduced what we call a tag. So the protein is completely normal, but there is a little thing attached that allows us to buy a commercial antibody and then follow the protein or purify it and stuff like that. So we don't have to make your own antibodies. But more importantly for this point is that we inserted these blue boxes here called FRT sites. And this is short for flipase recombinase targets. And what it does is that if we provide this flipase enzyme in the right tissue, it will basically delete everything in between these 2 blue boxes. And we can do this, again, in a tissue-specific manner, and this is awesome. So it's a completely different approach to essentially doing the same thing, namely deleting this gene. And the only difference between the two is the nature of the tag. We wanted to make sure that we don't introduce any artifacts due to the tag. So just to put this into context, this is probably the hardest CRISPR-Cas9 thing you can do, and this takes roughly a year from the original planning stage to getting the flies. So here's the same thing I just said in a more cartoon-like fashion. We replaced the normal copy of AGB with this copy, and when you then add flip by actually crossing it to another fly that makes this enzyme, you can cut out the gene. And we did this by a tissue-specific expression of this enzyme so that we can remove this gene again in the prothoracic gland. So long story short, did it repeat our porphyria phenotype, yes or no? And of course it did, because otherwise I wouldn't be talking about it. Okay, so here we go. That's the control, and then you get bigger ringlands and they shine in a beautiful red autofluorescent. So this confirmed our results, and now I believed it, I could sleep, and this was all good. All right, so I haven't told you what AGB is. And actually it's a well-studied gene, and it's linked to a human disease. But it has nothing to do with iron, at least not at this point. So what does it do? So it is an enzyme that puts branches on glycogen, of all things. There's a human version of the gene called GBE1. So it's very similar to the fly protein. It's 60% identical, and you can see the domain structure is also the same. And here in this cartoon, you can see how glycogen synthesis works. We don't care about all these details. But here in the last step here, you can see that these branches are attached. And if this doesn't happen, glycogen becomes so dense, it precipitates in the cell, it becomes toxic to the cell. So it's actually very dangerous. In fact, if you do not have a healthy copy of this gene, you are suffering from glycogen storage disease type 4 or Anderson disease. There's a milder adult form as well. And in Anderson disease, children typically die by the age of 5. Now, this is considered a rare disease. So 1 in 75,000 is sort of the normal occurrence, but there are 8 other glycogen storage diseases out there. So together, it's quite a common type of disorder. Now, this gene has not been linked to iron at all, but the only thing we could find was an ultrastructural study that found abnormal mitochondria in these patients. And recall that mitochondria is where most of the iron metabolism happens, heme biosynthesis, and actually also the assembly of these iron sulfur clusters. So that was something we thought was encouraging. Okay, so we have a glycogen enzyme and it has something novel to do with iron. What's going on? So we thought of 2 possibilities. The first one was, perhaps there's something wrong with iron delivery or iron transport. In other words, if there's not enough iron in the last step, right, to make heme, you could imagine that this creates a backlog of these fluorescent precursors, right? So iron would be an obvious choice here. And the other possibility was that perhaps this gene, AGBE, was affecting the expression or the function of these enzymes, and so these steps were disrupted. Now, you can test the last part easily, but we looked at the expression of these genes and they seemed normal. So we focused on iron right away, and that turned out to be the right track. So let me show you what we did next. So here is a survival chart. So what we do here is we take these 2 lines we talked about, the RNAi and the CRISPR, that both delete the AGBE gene specifically in the prothoracic gland. And we look at the survival. And we focus on the red here, which represents adults. You can see the RNAi line has roughly 4% survival to adulthood, but the CRISPR line is stronger with no surviving adults. Now, this is on normal fly food as you would get it from the fly kitchen, okay? Now we thought, why don't we put some iron in there and see if they do better? And that worked beautifully. So if you add iron, you suddenly get survival all the way to adulthood in a very significant manner. Okay, so that was very encouraging, linking this gene really to iron, that there was some kind of iron deficiency. Now that's survival. What about this autofluorescence? Let's take a look. So here's the autofluorescence I showed you before. This is our control, absent. And when you add iron to the medium, the fluorescence disappears. So we call that also a rescue. And again, these animals are fine. So clearly there's some kind of iron defect in these AGBE mutants. Okay, so how to proceed from here? There was nothing published, but there's a lot of stuff you can do by mining so-called databases. There are a whole bunch of studies that pour out data, proteomic data, genomic data, and these are not individual papers, but it's just data dumping, okay, from genome-wide efforts. In a human protein-protein interaction database, we found that the human glycogen branching enzyme was interacting with a protein called ACO1. And ACO1 is the official name of IRP1. And if you recall this, I showed you this slide. IRP1 sits at the center of cellular iron homeostasis. Okay? So it looked like that AGBE had something to do with this. And so suddenly we had to become experts in studying IRP1. But first of all, we wanted to know, is this true? Is this interaction, which we find in a database, in 1 of million of interactions, is this real? So we did this, we wanted to recapitulate this in our lab, and for this we used an approach called co-immunoprecipitation. So let me briefly show you how this works. So what you need here is a sample that contains essentially the proteins that you're interested in, namely what we call the bait protein and the prey protein. So what you want to know is the bait interacting with your prey? And what you need here are 2 antibodies. One against your bait. So if we look at the top here, so in this example, these 2 wouldn't interact. So when we pull down this blue protein here, it wouldn't be attached to anything because there is no interaction, right? Now, if there is an interaction, the orange protein is stuck to the blue, and we pull down this, we pull down the orange as well. Okay? And so now all we have to do is detect the orange protein by a second antibody. And because it's absent here, there's no detection, and as you will see this in the next slide, there would be no band because we resolved this in a gel, whereas there would be a band present here. And that tells you that the protein interacted. So let's take a look at the actual experiment. Again, human proteins, is this interaction real or not? This is done in cell culture, which is a little bit artificial to some degree, but I'll get into that. So there's a whole bunch of bands, so which one should you look at? Now, let's start with this control here, where we use an arbitrary protein, okay? And of course, the hope is with an arbitrary protein that it doesn't pull down your IRP1 protein. So that's the spot you need to look at, and you can see there's no band, maybe it's a little bit of a smudge there. So if we look at the equivalent position at the top, you can see a strong band, and that tells you that that GBE1 is indeed, at least in this system, interacting with IRP1. So we confirmed these data. That's great. But we're working on Drosophila, so let's do this in Drosophila. Well, we took it one step further. Recall that IRP1 can switch between this RNA binding form and the aconitase. So we wanted to know if there is an interaction, is there perhaps some specificity? Is it perhaps only interacting with this or with that? So how do we do that? That's not that easy. So we used some genetic tricks. So here's how we did it. So what we did is generating a bunch of, well, actually 3 different forms of this IRP1 gene. This is the Y type, the normal form. It can switch between the two. And perhaps you recall that this iron sulfur cluster, this here, needs cysteines to bind to. And we mutated this cysteine at position 450, and this is sufficient to abolish the binding of this iron sulfur cluster. And what happens then that this becomes constitutively RNA. All right. So this can switch. This becomes RNA binding and does not have a aconitase. That's lost. And here's the other way around. Here's some other point mutations that abrogate RNA binding but retain the aconitase activity. All right. So that's what we used. And so let's go back to or move on to this immunoprecipitation experiment. So at the bottom again, our controls, and we're going to look at these guys here, and they're all empty, which is good. So let's take a look at the first clone here, or the first construct. So this is the Y-type form, which can switch, and it does result in a band, so it's interacting. But the RNA binding form doesn't, whereas the aconitase form does. All right. So this is what we found. Okay. AGBE only interacts with the aconitase form. And this made no sense. Okay? This was contradicting everything we expected, because when you recall this paradigm, it's the RNA binding form that's doing all the iron regulation. People believe that the aconitase has nothing to do with iron regulation, and yet our novel iron regulator, AGBE, interacts with this guy. Okay? So that was cool. But we needed further validation. And for this, we used mass spec. And I'm going to rush through this technology. It's very similar to what I just described. But really, the difference is that we did this in the animal, in the fly. So this is more natural. Okay? And the way you should think about this is that any given protein has probably not just one partner, but multiple proteins it can interact at any given time. So in reality, there are multiple preys, and our bait, if we use an antibody, will pull down orange, green, purple, and yellow, and more. Now you do that, and what you get is a sample of all these interacting proteins, and we want to identify them. How do we do this? The answer is mass spec. Now, how does it work? Well, you take your sample, you run over to the chemistry department and say, give me the results. That's how it works. All right? Okay. But basically, if you want to know more, ask me later, but it's basically a very controlled digestion, and they can measure the mass of these peptides that result and look up in the library what those peptides are, and then they know what the protein is. So it's quite advanced. But the bottom line is you get back a list, and you can look at the list and see what has been interacting, what are these proteins interacting with IRP1 or AGBE or whatever. Okay, to make a long story short, we did a whole bunch of mass spec experiments in vivo, both with these knock-ins where we put in the tag, and also with so-called transgenes that make a lot more of these products. And we also chose tissues such as the prothoracic gland or the whole body, which is a whole lot easier because the prothoracic gland is tiny. So to give you an idea how tiny this is, to make a prothoracic gland sample working for mass spec, my student had to dissect 15,000 prothoracic glands over a course of 3 months. So that's why these here are ongoing, and it's a lot of work. Okay, what did we find? When we used AGBE as a bait, we pulled on IRP1A, hooray, confirming this happens in vivo. But we also did the opposite, using IRP1A as a bait, and both in the prothoracic gland and the whole body sample, it pulled on AGBE. Awesome. Worked beautifully. Now here, this is important. This was the guy who didn't interact. And what we did here, we produced lots of it. We should have picked up even small amounts that are interacting, but it wasn't on the list. So that's great. And if all this confused you, here's the executive summary. All right, so the aconitase form, also known as hollow IRP because it contains this cofactor, interacts with AGBE. The RNA binding form doesn't, and that was a huge surprise. So we wanted more evidence for this. And this was all protein-protein interaction, but we thought, is there a way we can do this using genetics? And so we used, again, a survival chart. So here is something I've showed you before. This is the CRISPR line where we remove AGBE in the prothoracic gland. They die. Okay. And back then I showed you adding iron rescues them. But here we do something different. Here we put back IRP1A back into this background, basically extra copies. We put in IRP1A and the form that can only bind RNA. And look what happens. When we put in the wild-type form, which can switch, we get beautiful rescue to adulthood. If we do this with the RNA binding form, it makes no difference to this, no survival. So this confirmed our protein-protein interaction data, or at least it was consistent with those data. And again, by now you know the drill. We're going to now look at the fluorescence, right? What happens to the porphyria phenotype? So we're going to start with the RNAi this time. So just mix it up a little bit. Here's our disruption of AGB alone. Now we put in the Y-type form, which is the guy that can switch, and we get beautiful disappearance back to normal, whereas the RNA binding form doesn't do that. So consistent with the survival data. Then we thought, all right, what about the aconitases? This is just the RNA binding form. Can the aconitase part rescue? And the answer is no, it cannot. So here's our 3R3Q form. And back then we didn't know whether this can act as an aconitase. So we used another aconitase from the yeast. Bottom line is, and the way you should interpret this is, you need both functions of this protein. You need the RNA binding form and the aconitase, suggesting the aconitase is essential. And we have then later gathered independent evidence that showed that the aconitase is critical. And then finally, we wanted to know, do the human's protein work in flies? And for that, we used human IRP1 and human IRP2. And there's a beautiful parallel to this result here, because the IRP1 in humans can switch and it rescues, the phenotype disappears. Whereas the IRP2 form is RNA binding, similar to this form, and it doesn't rescue, or not nearly as good. All right, so this tells you that AGBE and IRP1 kind of work in the same pathway, and that IRP1 is not properly functioning in AGBE, and that you can rescue it with IRP1. So that's great. But we now needed to take a look at what happens if you just interrupt IRP1 function. And so, therefore, we created a CRISPR-Cas9 mutant and removed the IRP1 gene in flies. There was no mutant available. And you can see that they die very early in development. They don't even make it to the third instar. But that's some normal fly food. If you add iron, they live fine, and you actually can keep them as a stock. So this is very similar to the AGBE mutant I showed you. Not only that, when you look at the prothoracic glands, they show in these two independent approaches also red fluorescence. So clearly IRP1A and AGBE have similar properties. Okay, so I mentioned that the aconitase is so important, but this whole isocitrate-citrate business didn't make any sense because it shouldn't be important. So the question was, is there some other undocumented function of this aconitase that nobody knew about? And the answer is yes. And here is the answer. So when we expressed IRP1A in the prothoracic gland, we noted something that we didn't understand. This protein went right to the nucleus. Now I should say that the classic paradigm states that this enzyme acts in the cytoplasm. Nobody ever mentioned anything about a nuclear function. Now what about the RNA binding form? This is one point mutation, and it can't enter nuclei. So that was very interesting. So in summary, what I've just shown you is that the single point mutation results in cytoplasmic distribution. And that's where an RNA binding protein should be, because it regulates messenger RNA. That happens in the cytoplasm. But the aconitase, surprisingly, was nuclear, and there also was some in the cytoplasm. So that was a huge surprise. So because I already told you and shown you that AGBE is interacting with the aconitase, which is shown here, we wanted to know whether this gene or protein was necessary for IRP1 or the aconitase to go into nuclei. And the answer is yes, and that's shown here. So we can stain the DNA with blue. That's called DAPI, and you see these nuclei, so you know where the nuclei are. And this is a stain for IRP1, and this time it's the knock-in, so it behaves almost, or it should behave like the endogenous gene. And you can see it's in a nuclei. But this is in a wild-type background where AGBE is functional. Take a look at what happens when we take out AGBE. The protein is in the cytoplasm. So AGBE is required for IRP1 to go in the nucleus. Why is that? Well, we're not entirely sure. But one theory could be that perhaps AGBE is a nuclear protein, and IRP1 sort of piggybacks onto it and goes in the nucleus. But that is unlikely because AGBE itself appears to be cytoplasmic. So you can see the blue here, and the green is outside the blue. So this suggests AGBE doesn't go into the nuclei. So we favor a different idea. We think that without AGBE, the IRP1 protein cannot form the hollow form, cannot become an aconitase. And without that, I've already shown you that you need the aconitase to go into the nucleus because the RNA binding form stays in the cytoplasm. And if that were true, this would mean that AGBE is somehow required for the maintenance or the insertion or stability of this iron-sulfur cluster. Okay. And that idea gained strong support when we went back to our mass spec data because we discovered another protein that supported this idea. Okay, so we've already seen this slide, and I used it to show you that AGBE is interacting with IRP1A. And we take a look at these 4 different conditions and look for proteins that occur on all lists and on all these conditions. There were only 2 proteins that were sort of surviving this. And one was a histone protein, and I will talk about this at the very end. And the other one was a protein called cis-D2. And cis-D2 is well known from vertebrates. And to explain its function, I need to backtrack a little bit. So we first have to figure out how does this iron-sulfur cluster gets into this IRP protein. And we know today that there's a whole bunch of proteins called the CIA. Yes, I'm working with the CIA, which stands for cytosolic iron sulfur assembly. And they make this compound and also insert it. And so that's the normal way by which a newly formed protein receives the iron sulfur cluster. But what happens is that this cluster is sensitive to oxidative damage and invariably will be ending up like this, and the whole protein will be essentially non-functional. And the CIA machinery cannot repair that. So the eukaryotic cells have a different system to repair this. And that's where Cyst-2 comes in. It actually has its own iron sulfur cluster and can donate this to client proteins, including IRP1, and swap it out and repair this. All right? So what I've just shown you in the mass spec data is that AGBE interacts with both of these proteins, and in fact, Cyst-2 also interacts with IRP1. They interact synergistically, as we have shown in co-IP experiments. And in the interest of time, I'm not going to show you that. So we think that AGBE is somehow involved in repairing damaged IRP1. And we are in the process of doing some experiments to actually show this directly. I'm going to show you one more experiment that suggests that these two work in the same pathway. And this is called the genetic interaction. It's quite elegant, actually. So what this is that we are using, again, this RNAi technology to disrupt the gene function. And we do this for IRP1A, IRP1B, and Cyst-2 in the prothoracic gland. And as you can see at the survival here, all these animals reach adulthood just fine. And there's also no autofluorescence, all right? But it gets interesting when you start combining these things. When you combine IRP1A and Cyst-2 here, you see dramatic collapse of the population with no adult survivors, no red here, and the appearance of this porphyria phenotype. So this tells you that these two genes work in the same pathway. And the way you can think about it is you have a pathway and you take away a little bit of one component, nothing happens. You take away the other component, nothing happens. But you do it together, the whole thing collapses. Okay. And the last one here is a nice control with IRP1B and Cyst-2, nothing happens. So it's not just a combination of two RNAi lines that triggers this. Okay, so the next thing we wanted to do is figure out, well, it's going in the nucleus, what it's doing there. Because I think we have talked enough about its role in repairing this oxidatively damaged protein. And so this is the last part of my talk. And so what is IRP doing in the nucleus? And to address this, we made these constructs where we, again, use the same constructs I already talked about. But here's the following idea. We use RNA-seq, which is short for RNA sequencing, and it's a technique to allow you to essentially measure the gene activity of all genes at once in a sample. Okay. And the way we did this is we expected that this RNA binding form, which you recall was only cytoplasmic, had essentially no effect in the nucleus because it didn't go there. Right? Whereas the wild-type form could do both, and this one should only really act in the nucleus because it couldn't bind RNA anymore. So we were hoping to see sort of a gradient where the nuclear effects would be strongest here and absent here. And that actually worked, which I couldn't believe, but it actually worked. And it's very hard to show you RNA-seq data in something that makes sense. So I'm going to show you a table that you cannot possibly understand or read. And it looks like this. But the point is, and I'm going to summarize the data just verbally, what we found was that the genes that were downregulated showed an interesting pattern. And that pattern was that it was enriched for genes that work in ecdysone biology. So all in blue here are ecdysone genes. In fact, 6 of the 7 Halloween genes were right in there. And the rest of the list reads like iron and heme biology. So when you do statistics and want to prove this, you can show that this list is enriched for heme, iron intense processes. All right. So, and now I'm going to enter briefly the realm of speculation because we wanted to understand how this can happen. How can this protein do this? And we will go back one more time to our mass spec data, this time in a slightly different format. So this is a slide showing you all the interactions we saw with the 3 different baits, shown in yellow here. And this is, of course, a busy slide, so I'm going to simplify it for you. Because what we were interested in is this. We want to find the proteins that are known to be in the nucleus, because that's where IRP1 was, and we were hoping to find some kind of clue of what the partners were in the nucleus. And you can see there are 4 proteins, all starting with H. And that's because they're all histone proteins. So histone 2A, histone 2B, and so forth. Now, I should mention that in this type of experiment, mass spec pull-downs, you get a lot of crap. And this crap consists of, not regular crap, but it consists of proteins that are highly abundant. So they stick to everything and you pull them down. And histones are certainly proteins that qualify as abundant. So when we first saw this, we thought this is not specific. But there are many reasons for us to believe that there is hope that this is in fact specific. So for starters, our controlled samples never showed any histones. And if you look closely, when we did this with IRP1B in 6 independent samples, we always pulled down these 2, never these. So there seemed to be some kind of specificity towards, or difference between the A and the B gene. Also, when you look at the human protein, there are 20 reported proteins, and one of them is a histone. And finally, these histone genes come in so-called clusters. So typically there are 20 to 30 histone 4 genes. But this variant here, H2AV, only comes in a single copy, and I would argue that this likely is a much less abundant histone protein. So again, argues for some kind of specificity. And so why is this interesting? Well, here's what we think could be going on as a working hypothesis. So in eukaryotes, when you want to activate a gene, and genes are, or the histones are invariably, as part of the process, acetylated. Okay? What this does, it opens up the chromatin and allows more access to transcription factors to turn on that gene. And this histone acetylation is done by a group of enzymes called histone acetyltransferases, or HATs. And these enzymes require a substrate, and that is acetyl-CoA. So what has this to do with IRP1? Well, one of the 2 main sources of acetyl-CoA is citrate, which is converted in the nucleus by ACL to acetyl-CoA. And as you recall, aconitases use citrate. So one working model would be that the aconitase form in the nucleus is basically depleting citrate by converting it to isocitrate, and therefore inhibiting gene activation by blocking histone acetylation. And that could happen in a local manner. Now, if you think that I've smoked too much weed in the recent time, it's like a crazy theory. It turns out that it's a hot topic trying to link metabolites to gene regulation. And so I pulled this from a Cell review from 2014 that basically illustrates what I just said. You would see here that citrate would be pumped out of the mitochondria and enter nuclei and then enter histone and the histone acetylation pathway. So this is something people know and people have looked at. All right, it's time to wrap all this up in a model and as a take-home message. Okay, at the center of my talk was this paradigm that has stood the test of time for 30 years, and that says that IRP1 can switch between the apo and holo form, depending on whether this iron-sulfur cluster is present or not. And people believe it's only the apo form that regulates iron through mRNA binding. The holo form, or the aconitase, is really to just diddle-daddle in the cytoplasm and convert the occasional citrate into isocitrate and vice versa. Now, I also mentioned that the CIA machinery is important for inserting this iron-sulfur cluster into the apo form and to generate this. All right. So I also showed you that CISD2 is important for maintaining or repairing oxidatively damaged IRP1A. And now I've shown you that AGBE is an important part of this because it interacts with these two on a protein-protein level. And importantly, what we think is going on is that when you mutate this gene, you essentially disrupt this repair process, and as a consequence of that, this accumulates, and these, over time, lose their function. And this then results, because you lose IRP1A in these red ringlands, that is this porphyria phenotype. And finally, there's belief that the aconitase has no function, and we think that's wrong. We think that it can, at least in some tissues, enter nuclei and regulate iron-intensive processes by a hitherto undefined mechanism. All right, I'll stop here and briefly thank the people who did the work. And most of the experiments you've seen today have been conducted by Nan. And the whole project was originally launched by Xu Shang. She worked on the original screen and the secondary screen, and Brian helped with the original screen as well. And Song and Satar work on other iron projects in the lab. I want to thank funding agencies, collaborators, and I want to thank you for not falling asleep in the dark light. And if you have any questions, let me know. Cheers.
Go for it. Okay, so an alternative idea occurred to me, and that is that because the aconitase originates from a protein that in fact has nucleic acid binding activity, is it possible that when the iron sulfur cluster forms, you're inactivating the RNA hairpin binding part, but converting it to something that could be a DNA binding part? And, you know,
well,
like would a chip analysis show this directly binding to DNA transcription factor?
Yeah, we definitely want to know whether, we know it's in the nucleus. What we don't know is it binding to chromatin or DNA, or does it bind to histones, or any of these combinations? The other possibility would be that it's just in the nucleoplasm and not binding to chromatin at all. So the first thing we need to do is validate whether we can pull down, there's a chromatin assay where we can pull down IRP and test whether it's binding to chromatin. That would be the first step. And I think chances are it's binding to specific modifications on histones and recognizing subsets of histones is my pet theory.
So the other thing is, can you make a mutant that has the enzymatic activity destroyed, but nonetheless still fulfills this regulatory role in the nucleus? Because that would tell you that it wasn't.
A non-aconitase but nuclear form?
Yeah.
For that, you would need a nuclear targeting signal hooked onto the C450S form, and we have done that. So we're making that.
Yeah, but it's getting in anyways with the help of the ATP,
no,
right?
If you want the aconitase intact or not?
I want the enzymatic activity broken.
Broken. So if you break that, you break the aconitase. The aconitase is the only form that we know of that goes in the nucleus. So you need to find another way to get it into the nucleus, and that would be a nuclear targeting signal.
Well, but you should be able to make an enzymatically inactive form that hasn't changed the conformation so well. You might be able to,
that's the thing. If it doesn't bind the cluster, there's a little video on the science paper who published this conformational change. It actually flaps out. It's like a huge change. So this cluster holds it all together. If you don't have the cluster, I don't see any other way of preventing this conformational change. So what we can do is
you want to keep the cluster present. You just want to change one of the amino acids so that the enzymatic activities.
But those are linked, so it's very hard to separate the two. So. Maya?
Ecdysone produced in the ovaries of adult insects?
Yeah.
Is there also iron metabolism going on?
We don't know, but I would bet yes.
So
I think.
Before 50s?
Well, first of all, every cell needs heme.
Yeah.
Every cell needs iron. So the answer is yes.
Yeah.
The question is whether it would be enriched in that tissue compared to other adult tissues. I don't know, but I would say, if I had to bet, I would say yes. Yeah.
Thanks.
Lisa?
I have a really simple question. Are there active uptake mechanisms for metabolites in nuclei?
Are there known uptake mechanisms? I don't know how these guys go through across the nuclear membrane, but they're small, so I imagine they go right through the pore. I don't know if there are any symporters or transporters that are specific to certain metabolites, or if it's just.
Would it have to be selective for citrate to get enough of a concentration to make a difference?
Yeah, I haven't looked into the specifics of transport. I actually don't know. You're right. If you want some kind of gradient, you want to make this, active and some kind of energy dependent process, but I actually don't know if that's been looked at. Yeah. Did you still have a question?
That's good. Do levels of dietary iron actually influence all these pathways?
Which pathways?
Taking iron out of the diet
Yeah.
and ending up.
So what we've done, well, if you don't have, okay, so you can experimentally take out, what you can do, you can add an iron chelator to the diet. So the iron is technically still in there, but surrounded by this chelator makes it unavailable to the fly. And when you do that, nothing happens initially. If you do that for 5 generations, the flies start dying. So they have an enormous repertoire of iron to begin with, and then you deplete this over the generations. And then they die, because you need iron to live. Right? The only known example that doesn't need iron is Borrelia burgdorferi, or whatever the name is, so the Lyme disease bacteria. There was a science paper showing that apparently it has iron, but it doesn't use it. So, yeah.
Sort of following up on Andy, what do you think would happen if you offered a normal Drosophila iron-enriched larva, iron-enriched versus the normal Drosophila versus one of your modified lines that has the need for extra iron? Do you think that the larvae can detect their lack of iron and choose one food over the other? Eating.
Oh, so you want to have a food
Choice
choice
experiment.
experiment? That goes basically back to taste, right? And it could be also selection
Taste
thing.
or experience. So I feel good after I
Now, I don't know anything about taste, to be honest. That would be an interesting experiment. The way we could we could dye the food and see whether they, end up having more of the dye than controls.
Or
Yeah.
divide your
Yeah. So the way we
food
would
into
do that,
chunks and
there's a
see.
food dye like bromophenol blue, and if we add that to the iron part, we could probably measure it to if it's equal iron or no iron. Maybe that's the way to do it. I don't think they would necessarily be able to taste it. At least not in pure iron form, is my guess. So I can't tell you. We haven't done that type of experiment.
Just wondering if they might associate eating that stuff with, feeling better.
Yeah. Well, you have to ask them.
You have to ask. Get one of your students who can set down 13,000 and do something like that for fun.
Yeah, get an idea how they feel. Any other questions? Yeah, Dave.
Possibly ecological ones for a change. Are there any Drosophila species that live on foods that are either not iron-rich or iron-poor? You might imagine that they would have evolved a different level of regulatory.
Asleen behind you. I only know of certain Drosophila species that live on cacti, but I've never seen anything with respect to iron levels.
You might imagine that doing a sort of looking at the phylogeny, you might find species in which this set points have changed, which might help you to understand a little
Sort of the
bit.
equilibrium has changed because they.
Right.
Well, I would
They
think
would
that.
either need to scavenge more, or
Yeah.
they would need to split it from their diet because of toxicity.
Well, I think that's why the system is there. So if a larvae are exposed to a low iron environment, they. And that's an interesting question. Can they sense that, or do they just, vanish from the face of the earth? Right, selection, natural selection. If they can taste that if there's some feedback mechanism that senses iron is low and then encourages them to go somewhere else, that would be cool. But
Right,
nobody
there
has
might
looked
be
at
cases
that.
where you're taking advantage of a low iron source, which would get rid of the competitors. But if you can evolve a way to scavenge all the iron that's there, you might be
Yeah.
done.
I don't know. Yeah.
Now you can travel to exotic places.
Yeah, Hawaii has 500 species. I'm sure I'll find something. Just one.
It's already done the
Iron
experiment.
uptake by organisms, though, is usually rather active. I mean, you've got ferritin and you've got various siderophores, so you need a pretty low iron environment to be so low that, because those are high affinity iron binders, right? So,
Yeah.
I mean, what do you know about the Drosophila organismal iron concentration from its food source? Because I imagine there's quite a system there for really actively extracting iron.
Well, the iron uptake in the gut is quite similar to what happens in vertebrates. So many of the main players that act in the gut are also conserved. So typically what happens is that the iron is taken up and then loaded onto a molecule called transferrin, and that's then released into the bloodstream in humans. And transferrin then docks onto cells that have the transferrin receptors. Now flies are a little bit odd because they have the transferrins, but they don't have the transferrin receptors. So it's something we have to find yet. So there's no ortholog for that. And all these reductases that change, ferric to ferrous iron and so forth, they are all conserved in flies and humans. So I would imagine that it's similar. Yeah. One of the major hormones that regulates systemic iron in humans is missing in flies. So we don't know what's taking over that role. So hephaestin is that. Any more questions? Well, if you want to know how I worked with the CIA, spied on Mike.
Nuclear secrets.
Thank you.
Yes, thank you.
I decided whether I was going to community this or not.
Kirst?
But
Yeah.